After studying this module, you shall be able to understand why nature has selected zinc as an integral component of metalloenzymes. We will also learn about the different kinds of zinc sites in proteins and will be able to identify them by looking at the zinc site.
Dear students, you are already familiar with the involvement of elements in biological system of the body. In this module, we shall learn about the biological role of zinc and also the classification of zinc sites in proteins. Zinc is the second most abundant trace metal in the human body after iron. It is essential for the activity of more than 300 enzymes covering all 6 classes of enzymes. Zinc in proteins can play an active catalytic role, generally as a strong Lewis acid, or it can act in regulatory or structural role, for example, maintaining protein structure and stability. Metalloenzyme is an enzyme which has a bound metal ion. There are several reasons for zinc to be an integral component of metalloenzymes. The electronic configuration of zinc 2 ion is argon core 3d10, and since it has a completely filled d shell orbital, its ligand field stabilization energy is 0, irrespective of the number of coordinated ligands, and hence no geometry is inherently more stable than the other. As a consequence, it readily forms low coordination complexes which have more acidity than high coordination complexes. Zinc can easily change coordination number from 4 to 5 to 6. Zinc 2 ions, being a D10 system, has no tendency to undergo redox reactions under biological conditions. Also, it is a good Lewis acid and can rapidly exchange ligands for rapid product dissociation. Unlike many other transition metals, zinc 2 ions not undergo hydrolysis at low pH to form hydroxo complexes. Zinc is known to a non-toxic element. In this module, we shall also understand the role of catalytic sites of zinc. There are basically 3 sites of zinc, that is catalytic, co-catalytic or structural site. In all cases, removal of bound zinc by a chelating agent can lead to the loss of enzymatic activity. In all catalytic sites, the function of zinc ion arises from its capacity to act as a Lewis acid. A zinc 2 ion which has a catalytic role in the enzyme is present at the active site of the enzyme and directly participates in the bond making or bond breaking step. The coordination sphere of the zinc ion is open and contains at least one water molecule along with 3 or 4 protein ligands. The co-catalytic sites occur only in the multi-metal zinc enzymes. Before the access structure detection of these enzymes, the function of these metal atoms were dubbed as modulating or regulatory. In a co-catalytic zinc site, there are 2 or more metal atoms, 1, 2 or all of which can be zinc. They are in close proximity to one another and operate as a functional unit. One metal plays the catalytic role and the other metal atom enhances the catalytic activity of the site. Examples of enzymes that contain co-catalytic zinc sites are alkaline phosphatase with zinc atoms and 1 magnesium atom, phospholipase with 3 zinc atoms, and nuclease P1 having 3 zinc atoms. Let's understand these concepts with the help of graphics and visuals.
We first try to understand the role of zinc in biology. Zinc is the second most abundant trace metal in the human body. The first zinc metalloenzyme, carbonic anhydrase 2, as shown in the figure, was discovered in 1940 by Keeling and Mann. The sphere in grey color is the zinc ion, coordinated to nitrogens of 3 histidine residues shown in yellow, and a water molecule shown in red. Since then, more than 300 zinc enzymes covering all 6 classes of enzymes have been discovered in different species. Zinc in proteins can play an active catalytic role, generally as a strong Lewis acid, or it can act in a regulatory or structural role in maintaining the protein structure and stability. As we have just seen, zinc plays a specific role in bioinorganic processes. This is because zinc has several distinct chemical features. Since it has a D10 configuration, LFCE is 0 irrespective of the number of coordinated ligands, and interconversion among its 4, 5 and 6 coordinate states is fast. This helps zinc to catalyze chemical transformations accompanied by changes in the metal coordination geometry. Dye-positive zinc ion does not participate in redox reactions. This lack of redox reactivity makes dye-positive zinc a stable ion in a biological medium whose potential is in constant flux. Zinc 2 plus is also kinetically labile, so that substrates and products can be readily introduced and removed. It is a good Lewis acid, especially in complexes with lower coordination numbers. It does not undergo hydrolysis at low pH to form hydroxo complexes and is non-toxic in nature. In this slide, we will learn about the different kinds of zinc sites in proteins and their characteristic features. Catalytic sites, co-catalytic sites, and structural sites are the three kinds of zinc binding sites in proteins. The catalytic site shown in this diagram is that of the enzyme thermolysine. The co-catalytic site is of the enzyme aromanase proteolytica aminopeptidase, and the structural site is of alcohol dehydrogenase. In the figure shown, H is a histidine residue, E is a glutamate amino acid residue, D is an aspartate amino acid residue, and SR is cysteine amino acid residue. In the catalytic sites, zinc binding geometry is typically distorted tetrahedral, sometimes trigonal bipyramidal, and zinc directly participates in the bond making or breaking step. Co-catalytic zinc sites occur only in multi-metal zinc enzymes. It has two or more metal atoms, one, two, or all of which can be zinc. One metal plays a catalytic role and the other metal atoms enhance the catalytic activity of the site. Zinc has a coordination number of 5 with slightly distorted trigonal bipyramidal geometry. In the enzyme aromanase proteolytica aminopeptidase, aspartate and glutamate act as bidentate ligands. A characteristic feature of co-catalytic site is the presence of a bridging ligand. In structural sites, zinc ion mainly stabilizes the tertiary structure, and the amino acid which is mostly coordinated to the zinc atom in these sites is cysteine.
The co-catalytic zinc sites. These occur only in multi-metal zinc enzymes. Before the X-ray structure detection of these enzymes, the functions of these metal atoms were dubbed as modulating or regulatory. In a co-catalytic zinc site, there are two or more metal atoms, one, two, or all of which can be zinc. They are in close proximity to one another and operate as a functional unit. One metal plays the catalytic role and the other metal atoms enhance the catalytic activity of the site. Examples of enzymes that contain co-catalytic zinc sites are alkaline phosphates with zinc atoms and one magnesium atom, phospholipase with 3 zinc atoms, nuclease P1 with 3 zinc atoms. In such zinc sites, a bridge is formed between the two participating metal atoms by a single amino acid residue like aspartate or glutamate, which binds simultaneously to the two atoms. Aspartate and glutamate have two electronegative oxygen atoms attached to carbon, hence they can easily serve as bridging ligands. In all the examples given above, one zinc atom is catalytic, while the remaining pair of metal atoms, zinc and zinc or magnesium, are linked to the bridging amino acid. The co-catalytic metal atom site differs from the catalytic site in the kind of the ligands, number of the ligands, and hence geometry of the complexes, as well as the presence of the bridging ligand in co-catalytic site. The catalytic site, zinc site, contains generally histidine. The co-catalytic zinc site have aspartate and histidine. Some multi-zinc enzymes have serine, threonine, lysine, and tryptophan as ligands in the co-catalytic zinc sites. The common coordination number of zinc in catalytic site is 4 with geometry distorted tetrahedral, whereas zinc in co-catalytic site have coordination number of 5 with geometry of slightly distorted trigonal bipyramidal. The spacing between the ligands in co-catalytic zinc site is irregular. Structural zinc sites. Zinc can play a structural role in certain proteins. In structural zinc sites, the zinc ion mainly stabilizes the tertiary structure. It has a symmetrical tetrahedral geometry satisfied by 4 amino acid side chains. There is no solvent coordinated to the metal. The amino acid, which is mostly coordinated to the zinc atom in these sites, is cysteine, with histidine also being present in many cases. The spacing between the coordinate ligands is irregular. A zinc finger is a small protein structural motif in which coordinated zinc plays a structural role contributing to the stability of the motif. Zinc fingers are structurally diverse and are present among proteins that perform a broad range of functions in various cellular processes such as replication and repair, transcriptions and translation, metabolism and signaling, cell proliferation and apoptosis. Aspartate carbamyltransferase is the only enzyme containing only a structural zinc atom. Alcohol dehydrogenase has 2 zinc atoms, one a catalytic and other a structural zinc atom. 4 cysteines coordinate the structural zinc in a tetrahedral geometry. Let's have a look at the 3D structure of alcohol dehydrogenate.
As we have seen in the previous slide that in the catalytic site, the zinc ion has at least one water molecule coordinated to it. The bound water can be either ionized to zinc-bound hydroxide, polarized by a general base to generate a nucleophile for catalysis, or be displaced by the substrate. The pK of metal-free water is 15.7, but can be reduced to 10 in hexaacco zinc-2 ion and 7 in a complex of zinc which has 3 nitrogen donors. This allows for facile ionization of water. Now that you're familiar with the role of zinc-bound water, we will now try to understand broadly the mechanisms for the catalytic action of zinc enzymes. There are 2 limiting cases. In the zinc hydroxide mechanism, the hydroxide produced by ionization of bound water acts as a nucleophile and attacks the carbonyl carbon of the substrate. In the zinc carbonyl mechanism, the zinc ion acts as a Lewis acid and the substrate attaches to the zinc ion through a carbonyl group. In this module, you have learned that zinc in proteins can play an active catalytic role, generally as a strong Lewis acid, or it can act in a regulatory or structural role. Zinc may be present at a catalytic, co-catalytic, or structural site. The different zinc sites differ in the kind of ligands, number of ligands, and hence the geometry of the complex. And finally, 2 broad mechanisms for catalytic action of zinc enzymes are the zinc hydroxide mechanism and the zinc carbonyl mechanism.
Let's now summarize what we have learned in this module. We have understand the role of zinc in biological system. That is, in protein, zinc plays an active catalytic role. Zinc may be present at a catalytic, co-catalytic or structural site. In catalytic sites, the function of zinc ion arises from its capacity to act as a Lewis acid, and in co-catalytic sites occur only in the multi-metal zinc enzymes. In structural zinc sites, zinc ion mainly stabilizes the tertiary structure. We have also studied that zinc has a symmetrical tetrahedral geometry satisfied by 4 amino acid side chains. There is no solvent coordinated to the metal. Amino acid, which is mostly coordinated to the zinc atom in these sites, is cysteine, with histidine also being present in many cases. The spacing between the coordinate ligands is irregular. The different zinc sites differ in the kind of the ligands, number of the ligands, and hence geometry of the complex. We have also understand the catalytic action of the coordinated water molecule, that is the bound water can be either ionized to zinc coordinated hydroxide or polarized by a base from a nearby amino acid of enzyme or be replaced by the substrate. The preferred pathway for activity water is determined by the identity of the other 3 ligands and their spacing. The 2 general mechanistic pathways have been proposed for the catalytic action of the zinc enzyme. In the zinc hydroxide mechanism, the hydroxide produced acts as a nucleophile that can attack the carbonyl carbon atom. In the zinc carbonyl mechanism, the substrate attaches to the zinc ion through a carbonyl group, which is then polarized, indicating a nucleophilic attack at the carbon atom of the carbonyl group. In the next 2 modules, we shall also discuss the structure, active site and reactions of the 2 zinc proteins, that is carboxypeptidase A and carbonic anhydrase 2.